Hello students, hope you are fine. We are here again with a new activity. So today's activity is to prepare a stained temporary mount of human chick cell. Okay, and we are going to observe the different components of these cells. Apparatus required for this is a clean glass slide. Why we use this clean glass slide? We have to place or pour our sample over it, and we have to cover it with the cover slip. Toothpick is needed to scrape the material from the inner lining of the mouth. Next, the dropper functionally it is used as to spread the material or to receive or to drop the material. Compound microscope, obviously the main, and the methylene blue stain. Why we use the methylene blue stain? Methylene has one property to make the bonds with the genetic material, that is the DNA and RNA. Okay, now we will, while using this methyl blue and observing under the microscope, we will see the dense colored structure, that is the nucleus, and the dense color is due to the methylene blue. Okay, now the glycerin we use it just to make the air free environment, bubble free environment, blotting paper and distilled water. These are the material required to perform the today's activity. Now how we will going to perform this? First of all, for the sample we have to scrape the material from the inner lining of the mouth. Then we have to rinse our mouth very gently, clearly, and then with the help of toothpick we have to scrape the material and we have to place over the slide. After that we have to add few drops of water or a weak solution of methylene blue. Then finally after mounting we have to use the methylene blue so that and we will keep it for 1 to 3 minutes so that the dye will form a complete or a good bond between the genetic material. Then we will place the cover slip and we will remove the excess of dye, excess of water from the slide, and we will see under the microscope, and definitely we will find a good result. So let's move to the lab and perform this activity. Okay students, now we are in our laboratory and we are going to perform activity to observe the cells by preparing a temporary amount of stained human chick cell. So the material required is, first of all we have the methylene blue, we have the glycerin, this is our slides, we have the cover slip, we have this dropper, and we have this watch glass to keep the sample and other things, and we have the toothpick as scraper. Here we have the distilled water. So with the help of these materials, let's start our experiment to observe the epithelial cells under the microscope. So first of all what we have to do, we have to rinse the mouth gently. Okay, now after that we have to scrape or we have to take the cells from the inner lining of the mouth. Here we are taking the cells or the material from inner lining of the mouth. Now okay, we have to place this material over the slide and we have to pour a little of the water. Okay,
remove that.
now we have spreaded this material. Okay, next we will keep it for 1 minute. Okay, now this is our dye, that is the methylene blue. Now we have to drop
Take one drop.
this dye over the slide or over the sample. Okay, make sure that we should not use very concentrated dye, or we should not use very excess of dye. Now we will keep it for 2 to 3 minutes so that the methylene blue will make a bonding with the desired observation or material which we want to observe under the microscope. Now rinse the extra dye from the slide. Okay, we have gently rinsed the extra dye. Now we will use 1 drop of glycerin. Here we have used the 1 drop of the glycerin. Okay, now just wait for a little bit, 2 to 3 minutes, and then we are going to place the cover slip. This is the cover slip, and I am putting it gently over the sample. Now I am just knocking it so that the air bubbles may be removed from here. Okay, now we have placed the slide over the blotting paper so that excess spreaded methylene blue and any other solution like water, glycerin, they are removed from the slide. Now it's very clear. Now we will keep it for 2 to 3 minutes. After that we will observe under microscope. Okay, we will observe under the microscope. Please take a clear picture. Okay, dear students, what you are watching is our epithelial cell, and the dense colored particle is our nucleus. So I hope you got the core concept. Now with the help of your teacher, you please perform this experiment in your labs. Thank you. Have a nice day. Okay, coming back from the lab, and with what we have observed under the microscope is a large number of flat irregular shaped epithelial cells. Okay, our motive was to see these epithelial cells. Definitely you have not observed the cell wall, and we have observed a dense structure, densely stained structure, and that structure is known as the nucleus. So that's all for the today's activity. Hope you have understand and enjoyed it a lot. Thank you.